Okay, guys, so this is section 7.1. It's cell discovery and theory. So, the history of the cell theory, there's a lot of dates here and a lot of people. Really, the two that you really need to know are Robert Hooke and Leeuwenhoek. So, Robert Hooke made a simple microscope and he called his observations cellulae, which is the Latin term for small rooms. So from Robert Hooke's term, we have our term known as a cell, and a cell is a basic structural and functional unit of all living organisms. Then in 1683, Leeuwenhoek discovered single-celled animal-like organisms now called protozoans. Now, as we go on down the timeline, in 1838, it was concluded that all plants are composed of cells. In 1839, it was reported that animal tissues are also composed of individual cells. And then in 1855, it was proposed that all cells are produced from the division of existing cells. So all of these things together developed what we know as the cell theory. So, the cell theory is one of the fundamental ideas of modern biology, and it includes three principles. One is all living things are composed of one or more cells. Two, cells are the basic unit of structure and organization of all living organisms. And three, cells only arise from previously existing cells, with cells passing copies of their genetic material onto their daughter cells. So basically, in a nutshell, right, all living things are composed of one or more cells. Cells are those basic structures for living organisms, and then a cell comes from a pre-existing cell. We don't just have a cell out of thin air. It has been, the genetic information found in that cell has been passed on from cell to cell. So there are different type of microscopes that we need to talk about. Microscopes are how we look at cells, right? Cells are too small to see with the naked eye, and so we have to use microscopes. So there are two categories of microscopes. There are compound microscopes and electron microscopes. So talking about compound microscopes first, compound light microscopes consist of a series of glass lenses and use visible light to produce a magnified image. Each lens in the series magnifies the image of previous lens. So, for example, we have the two lenses, and individually they are 10 times. The total magnification would be 100 times because you multiply 10 times 10. Scientists will often stain cells with dye to see them better, and the maximum magnification without blurring the subject in a compound light microscope is 1,000 times. So just to kind of show you, so here's the picture, right? Right here is where the light comes from. It shines up onto our slide, and these are our lenses. So these here are 10, okay? So these are the ones that are different sizes. You have a 10 times, a 40 times, and a 100 times. So it's going to be, as you see here, it's saying if you're looking at two lenses individually magnified 10 times, well, that's talking about this is 10, and then this is 10. Okay, so that's what 100 would be. But if we flipped it, then it would be 10 times 40. Or if we flipped it again, it would be 10 times 100, which is where the 1,000 maximum magnification comes into play. So keep in mind that these lenses are always 10, okay? These are the ones that you're going to change, and that's why it multiplies by 10 every time. So with electron microscopes, these were developed in the 1940s. Instead of lenses, these use magnets to aim a beam of electrons at thin slices of cells. So remember, we talked about electrons a lot last chapter. Those are negatively charged particles. So these they use magnets to aim beams of electrons at thin slices of cells to magnify the specimen. There are three types. There's TEM, SEM, and STM. So talking about each one of those more specifically, a TEM is a transmission electron microscope. It's called this because electrons are passed through a specimen to a fluorescent screen. Thick parts of the specimen absorb more electrons than thin parts, which forms a black and white shaded image of the specimen. So that's what this is here. So this is what the TEM looks like. And electrons are passed through the specimen onto a screen. And this is what would show up on the screen. So notice how the thicker parts of the specimen are shaded darker because more electrons have been absorbed there versus these lighter parts. TEMs can magnify up to 500,000 times, but the key with these are the specimen must be dead, sliced very thin, and stained with heavy metals. So a compound light microscope, we can look at something living under there, whereas with a TEM, it can only observe non-living cells and tissues, right? So something dead. So an SEM is a scanning electron microscope, and this directs electrons over the surface of the specimen, producing a 3D image. And like the TEM, this can only observe non-living cells and tissues. So here's a picture of an SEM. It directs electrons over the surface of the specimen that it's viewing, and it produces this 3D image. So we have an STM. This stands for scanning tunneling microscope. This involves bringing the charged tip of a probe extremely close to the specimen so that the electrons tunnel through the small gap between the specimen and the tip. So the tip is charged, and it causes the electrons to go through this tunnel over the specimen, and this also produces a 3D image. This produces a 3D image on a computer, but it allows us to view things as small as what an atom would do. So that's what this is a picture of. This is from an STM image. It is of DNA. So you can see there are cracks and depressions that appear darker, and raised areas appear lighter. So unlike a TEM and an SEM, STM can be used with live specimens. Things don't have to be dead. And here's a picture of what that microscope looks like. So here's where the bulk of this chapter is going to come from. Okay, there are basic types of cells, and there are different types of cells, but all cells have at least one physical trait in common, and it's that they have a plasma membrane. Now, a plasma membrane is a special boundary that helps control what enters and leaves the body. Okay, so that's found on the outside of the cell. There are two types of cells, or two categories of cells. They're prokaryotic and eukaryotic. Generally, eukaryotic cells are 100 times larger than what prokaryotic cells would be. So we're going to look at a TEM photomicrograph of these two cells. On the left, we have a prokaryotic cell, and on the right, we have a eukaryotic cell. Now, here you can't really tell that a eukaryotic cell is so much bigger than what a prokaryotic cell would be, just because they're both magnified. But eukaryotic cells are, like the previous slide said, generally 100 times greater than what a prokaryotic cell would be. And that's because eukaryotic cells have these organelles found within it, whereas a prokaryotic cell does not. So those two categories, prokaryotic and eukaryotic, are based on internal structures. So we said that all cells have plasma membranes in common, right? So both have a plasma membrane, but one of those cells contain many distinct internal structures called organelles, right? So that eukaryotic cell has organelles. Organelles are specialized structures that carry out specific cell functions. And so later in this chapter, we're going to talk about each organelle specifically, but we're going to take a little closer look at the two categories. So we have eukaryotic cells. Eukaryotic cells contain a nucleus and other organelles that are bound by membranes. And you'll hear organelles considered or referred to as membrane-bound organelles a lot. The nucleus is the distinct central organelle that contains the cell's genetic material in the form of DNA. So the nucleus is going to be something that distincts a eukaryotic cell from a prokaryotic cell, because a prokaryotic cell does not have one. Only a eukaryotic cell has a nucleus. So it's that distinct central organelle, but it's important because it contains the cell's genetic material in the form of DNA. Organelles enable cell functions to take place in different parts of the cell at the same time. So each organelle has a specific function, and they can all take place at the same time, right? So the nucleus holds the genetic material that can do that at the same time as what the plasma membrane is keeping things from coming in and out of the cell. Most organisms that are made up of eukaryotic cells are called eukaryotes. Okay, so humans, animals, plants, things like that are considered to be eukaryotes. There are some unicellular organisms, such as algae and yeast, that are also considered eukaryotes. Prokaryotic cells, however, are defined as cells without a nucleus or other membrane organelles. So most unicellular organelles, now remember, we said some, like yeast and algae, are considered eukaryotic, but most unicellular organisms, such as bacteria, are prokaryotic cells, and they're called prokaryotes. So tomorrow we're going to take a little closer look at the difference between eukaryotic and prokaryotic cells. But the big thing that you need to understand is that a eukaryotic cell has a nucleus and a prokaryotic cell does not. That's going to be the one defining thing. There are going to be several things that you can tell the difference, because eukaryotic cells have several organelles. But the nucleus is the defining one that a prokaryote would not have versus a eukaryote having it.